We'll demystify CRISPR, how a bacterial immune system became a precise genome editing toolkit. You will learn the molecular steps behind Cas proteins, the repair outcomes that determine edits, real-world applications, current limits, and ethical implications, origins and foundations, DNA, CRISPR arrays, and Cas proteins. CRISPR began as a curiosity. Repetitive DNA sequences discovered in 1987 in bacteria that later proved to be molecular records of past viral infections. Microbes store short spaces, snippets of phage DNA, between repeats, and use CRISPR-associated Cas proteins to recognize and destroy matching invaders. The CRISPR-Cas9 system, popularized for genome editing, originates from Streptococcus pyogenes. SpCas9 is a single protein of about 1,368 amino acids guided by RNA. In 2012, Jennifer Doudna and Emmanuel Charpentier showed that a programmable 20-nucleotide guide RNA could direct Cas9 to cut DNA, a discovery honored by the 2020 Nobel Prize in Chemistry. Mechanism: how CRISPR-Cas9 finds, cuts, and relies on cellular repair. A CRISPR edit depends on two molecular parts: a guide RNA, typically 20 nucleotides matching the target, and a Cas nuclease. Cas9 recognizes a short PAM motif. SpCas9's PAM is NGG, 2 to 3 bases, then forms a DNA-RNA hybrid and cleaves both strands roughly 3 bases upstream of the PAM. The cell repairs that double-strand break by non-homologous end joining NHEJ, which often produces small insertions, deletions of 1 to 10 base pairs, or by homology-directed repair HDR, if a donor template is provided. HDR efficiencies commonly range below 10 to 30% in many cell types, which makes precise edits more challenging. Newer tools like base editors, 4 to 5 NT activity window, and prime editing reduce reliance on HDR. Applications: medicine, agriculture, and diagnostics. CRISPR has moved rapidly from bench to application. In medicine, ex vivo CRISPR edits of hematopoietic stem cells have entered clinical trials for sickle cell disease and beta thalassemia. Candidate CTX001 reported durable increases in functional hemoglobin in trial participants. In agriculture, gene-edited crops such as non-browning mushrooms were developed and in some jurisdictions treated differently from transgenic GMOs. CRISPR-based diagnostics, Cas12, Cas13, enable rapid detection of viral RNA and were adapted for SARS-CoV-2 research assays. Other directions include gene drives to control vector populations and animal model generation, each with technical and ecological caveats. Advanced topics and implications: delivery, safety, and ethics. Key practical barriers remain. Delivering CRISPR in vivo requires vectors. Adeno-associated virus, AAV, vectors have 4.7 kb packaging limits, which challenges delivery of large nucleuses like SpCas9 and often requires splitting or using smaller Cas variants. Off-target editing rates vary with guide design and cell type. Rigorous validation, deep sequencing, is essential. Newer editors, base editors, and prime editors lower double-strand break frequency and expand edit types, but raise their own off-target profiles. Ethically, somatic therapies are widely pursued, while human germline editing is globally contentious after the 2018 He Jiankui case. Regulatory frameworks and public dialogue are critical for responsible deployment. CRISPR transformed a microbial immune strategy into a versatile genome editing toolbox. Understanding Cas targeting, cellular repair outcomes, delivery constraints, and ethical boundaries clarifies both the extraordinary potential, curing genetic disease, improving crops, and the technical and social responsibilities that accompany it.